Distinguished colleagues, it has been a great privilege to me to preside over this conference, and I present to you my presentation overview report, forage resources and biological characteristics of honeybees, Apis mellifera, in the Perm region. Honey production capacity is one of the natural forest resources. It is also a trophic factor for bees. Sustainable beekeeping depends on the amount of honey plant resources, subspecies of honeybee plants, and the selection of honeybee subspecies for breeding. Dark forest honeybee Apis mellifera mellifera shows the highest honey productivity under natural forest conditions in Perm region. Honeybee subspecies characteristics include not only external traits, but also internal ones, such as genotype and enzyme analysis. The aim of the work was to analyze the honeybee forage resources in Perm region to identify the subspecies composition of honeybee, emphasizing the enzymatic biological characteristics of purebred bees. In the course of the research, the following results or conclusions were observed. 1. The species composition of honey plants was determined. The honey reserve of forest honey plants in Perm region was estimated. Next, the morphometric and tautological parameters of honeybee colonies of the Apis mellifera mellifera and Apis mellifera carpatica were studied. Next, the biochemical traits of the studied bees in winter period were examined. Next, the honey yield from tested colonies were estimated. Next, economic efficiency of using honeybee forage in Perm region and the future perspective were determined. The studies were conducted from 2003 to 2010 years in the periods of Perm region. Perm region is a part of the territory of Russia. It has its own features of relief, climate, and types of honey plants. Perm region is the northern boundary of honeybee distribution. Honeybee have acquired a whole set of adaptation characteristics to survive under this condition. Honeybees are adapted to local climate. For example, characteristics. More than 70% of Perm region are forest lands. So the main honey plants are of forest ecosystems. Next, honey collection period is very short, 1 month, July. The main honey plants are willow, herb, and linden. Next, there is low nectar secretion in September. Next, winter period is 6 months from October to April. In winter, bees cannot fly. They are kept in winter hives in the apiary. Next, honey harvesting time in spring is unstable. It lasts for a week or two in May. The main honey plant is willow. Honeybee disease is nosema and toxicosis. Studies were carried out in accordance with the basic requirements for the design of experiment in beekeeping developed by the Bee Breeding Research Institute, Moscow, Timorazov Agricultural Academy. In accordance with the aims and objective of the work, the following parameters were determined: species composition of honeybee plants, honey stock of the area. Biological traits include etological and morphometric parameters. Etological parameters include behavior responses of bees and copcaping. Breed purity of honeybee was determined by the method for measuring the exterior features of honeybees. The following parameters were measured: proboscis length, the length and width of the right forewing, cubital index, the length and the width of target 3, Vox mirror. Commercially available traits of the honeybees are honey productivity of colonies, their strength and overwintering. Honey productivity was determined by weighting the control colonies during the main honey production period. The study of overwintering was based on the activity of catalase and phosphatase as an indicator of heat production. Selection of honeybees for enzymatic analysis was carried out by random sampling in the central areas of Peru region. A total of 1,422 samples of Apis mellifera mellifera and 334 samples of Apis mellifera carpatica were examined. Storage of samples and preparation of honeybee gluconide were carried out in accordance with relevant recommendations. Enzyme activity were measured with the use of spectrophotometer for catalase and photoelectric colorimeter for phosphatase. The change in the transformation rate of the substrate model, pyranitrophenol for phosphatase and hydrogen peroxide for catalase was measured. Studies on enzyme activities were carried out in the laboratory of the Institute of Ecology and Genetics of Microorganisms in Perm. Through photometric methods are used in studying enzymatic characteristics of living organisms of different subspecies, but they are used for the first time in the study of enzyme activity in bees. Descriptive statistics were used to describe the research data in our study according with the recommendations. To study the honey stock of the area we used to scheme the forest vegetation zoning in Perm region, we can speak about 4 forest zones: zone of mountain forests of the central Urals, zone of middle taiga, zone of southern taiga, and zone of mixed forests. The time of blooming of honey plants depends from the zone. Honey plants in the zone of mixed forests bloom first. The same honey plants start to bloom in 5 days in the southern taiga and definitely 5 days later in the other zones. You can see figure 3. Melliferous capacity of honey plants in per region per blooming time, tones. The following results were obtained. The graph shows that early spring is the time for collecting honey in the forests of middle taiga. There are blooming of willow. The second half of spring and early summer is the time for collecting honey in all forest land, except the middle taiga zone. The nectar production of honey plants is reduced due to the wind or weather conditions. The period of the second half of summer is also called the main honey flow. Honey stock of this period is amount to 70% of the total stock. A significant amount of honey productivity during the second half of summer is provided by the mass blooming of linden, 66.5%, and you see in picture. Also, melliferous capacity includes nectar secretion of honeysuckle, shrubs, willow, mountain ash, rose hips. This shows the highest melliferous capacity comparing to other plants. The remaining 7.4% of melliferous capacity is due to different types of honey plant species. According to the results of the long-term observation in the area of mixed forests, willow blooms on April 20 and flowering lasts for 30 days for all species of willow. Mountain ash and honeysuckle begin to bloom on May 25 with the blooming period of 14 days. Dustiness blooms from July 2 for 20 days. The linden blooms from June 23 for 14 days. The indicated blooming period may shift due to the weather condition of a particular year of study. The total honey plant capacity as to each region allows us to estimate the honey stock of forest lands of Perm-Khyber regions. Honey stock of the mixed forest zone is 37%, southern taiga 35%, mountain forests 19%. The only honey collection of the early spring is in the middle taiga zone, though insignificant in size, 9%. Along with honey plants, the breed of honeybees is also important. Morphometric parameters most commonly used in honeybee breeding are proboscis length, wings length, and width cubital index. There are hereditary persistent traits that clearly characterize the breed of bees. All studied morphometric parameters of honeybees, Apis mellifera mellifera, Apis mellifera capatica, meet this standard. According to the obtained morphometric data and beekeepers' data in Perm region were revealed 52% of Apis mellifera mellifera, 40% of crossbred honeybees, 8% of Apis mellifera capatica. The main commercially valuable traits of honeybee colonies in Perm region were studied. The Apis mellifera mellifera colony strange before the main honey flow period was 25.6 seams between combs. That is 1.2 seams more than the strength of Apis mellifera capatica. When expressed in mass units, the strength of the Apis mellifera mellifera honeybee colony is 300 grams more than strength of the Apis mellifera capatica honeybee colony. The analysis of the control beehives of honeybee colonies showed that the gross honeybee yield in Apis mellifera mellifera was 4 kilograms higher than the one of the Apis mellifera capatica. The amount of marketable honey received from honeybee colonies was 40 and 40.8 kilograms for Apis mellifera mellifera and Apis mellifera capatica respectively. During the active period, weather conditions contribute to the development of the colony Apis mellifera mellifera, but not Apis mellifera capatica. By the period of the short and intense main flow of honey, the colony strange, Apis mellifera mellifera was maximum, which subsequently led to a greater number of bees ready for overwintering. Ethological characteristics of honeybee colonies of Apis mellifera mellifera and Apis mellifera capatica in Perm region were studied. The Apis mellifera mellifera bees are higher aggressive. During examination, bees were restlessly running around on the frame, unlike the Apis mellifera capatica bees, which continued their walk quietly. Both breeds had honeycomb with dry keeping. In the course of the study, we built a diagram characterizing the annual life cycle of honeybees Apis mellifera mellifera in Perm region. Winter or flightless period lasts 220 days, that is more than 6 months, from October till early April. The growing season in the area lasts from 145 to 165 days. That corresponds to about 5 months. The average daily temperature during this period is above +5 degrees. Long flightless winter period, more than 6 months, and short period of honey collection are the main conditions of the development and survival of honeybee Apis mellifera mellifera in the region. Winter hardiness is one of the most important characteristics of Apis mellifera mellifera. The study of winter hardiness of honeybee Apis mellifera mellifera according to the energy needs of the body was carried out. Phosphatase is an enzyme responsible for body energy demands. While analyzing the results of phosphatase activity, its mean body energy demands, maximum and minimum demands of honeybees during the winter period were revealed. Increase in energy demands in February is evident. It is necessary to maintain a constant temperature regime in the hive due to lower external temperatures. The next enzyme studied, also an indicator of winter hardiness, is catalase. Prevents poisoning of the body by metabolic products. For example, catalase helps to keep the excrement accumulated in the intestines of honeybees over a long winter period. Figure 9 shows the extreme parameters of catalase activity and the interval forms by them. The narrower the catalase activity range, the higher the winter hardiness of honeybee colonies. In March, the diversity of values increases, which is typical for the end of the winter period. Honeybees are preparing for brood rearing. The figure clearly shows the overall decrease in catalase activity, which is consistent with data relating to a decrease in catalase activity during honeybee aging. In the course of study, we compared Apis mellifera mellifera with Apis mellifera carpatica at the end of the flightless period. Analysis of phosphatase activities in bees showed their division into 3 groups typical for both species. However, high energy metabolism of Apis mellifera carpatica is 1.5 times significantly higher than the similar values for Apis mellifera mellifera. The energy metabolism of Apis mellifera carpatica with average parameters is 2.9 times higher than the corresponding values for Apis mellifera mellifera. A comparison of low parameters shows a difference of 10.1 times with the predominance of energy metabolism in Apis mellifera carpatica. Thus, the energy metabolism of Apis mellifera carpatica is higher than that of Apis mellifera mellifera, which indicates that the honeybees have aroused from winter dormancy. In condition of permeridian, this trait of Apis mellifera carpatica has a negative effect on the winter hardiness of colony. Catalase, called preserving enzymes, has a crucial role for winter hardiness. Comparison of different honeybee subspecies revealed the different status of honeybee colonies. When the Apis mellifera mellifera honeybee colony remains statistically unified groups, the Apis mellifera carpatica honeybee colony divides into 2 groups. In this case, the Apis mellifera carpatica group with higher catalase activity has significantly different values, which allows us to consider the Carpathian bees to be arousing from diapause. Thus, the activity of catalase increases with the development of post-diapause condition. The study of theoretical honey stock of forest lands in Permeridian revealed 390,919.3 of honey available to bees. This amount of honey is sufficient for having more than 3,007,000 honeybee colonies. To date, according to the statistics, there are less than 200,000 bee colonies in per region. Thus, only 6.7% of honey resources of the region is used. As a result, the economic losses associated with the underproduction of honey amount to 153,967.7 million rubles. Allow me not to detail on the findings, as I've already talked about them during my presentation. Honey producing potential of the forest lands in Perm region is about so many tons of honey. According to this, we would recommend to consider the possibility of increasing the number of honeybee colonies up to 3,007,200 honey bee colonies. We would also recommend to take into account biochemical parameters, phosphatase and catalase activity when selecting breeding colonies. The author is grateful to Maximov Alexander Yurievich and employers of the Laboratory of Chemical Mutagenesis for providing the basis for the enzyme analysis and consultation of all methodological issues. Former students of the Faculty of Biology and Chemistry, Elena Bessonova, Svetlana Lakina, for participating in enzymes analysis. Also, Sipadova Nina Igorevna for faith and support, Litvinov Nikolai Antonovich here, Department of Biology, for this assistance in the processing of digital data. Also, thanks for my family. I thank everyone who wrote the reviews and those who are present there today, and they will help to defend my work. Thank you for attention.